Scattered throughout the thylakoid membranes, millions of photosynthetic units capture the energy of the sun in the light reaction process. Each photosynthetic unit consists of two photosystems, called P680 and P700. Green pigment chlorophyll A molecules in both P680 and P700 absorb photon energy. Chlorophyll A absorbs a photon directly if the photon has just the right amount of energy. If the photon doesn't have the exact energy required by chlorophyll A, then it is the responsibility of the antenna pigments around the chlorophyll A to absorb the energy and pass it on. As photons continuously bombard pigment molecules, it is the double bonds of the molecules that take the brunt of the bombardment. A photon strikes one of the electrons in a double bond, jolting the electron from its orbit. In order to continue absorbing photons, the molecule which has become electron deficient must constantly replace the lost electrons. These replacement electrons come from water, readily available within the thylakoid. In a process called photolysis, water molecules split, yielding two electrons, two hydrogen ions, or protons, and one atom of oxygen, which quickly bonds with a second oxygen atom to form one molecule of oxygen gas. Most of the oxygen is released into the atmosphere. The protons drift into the interior of the thylakoid, and the electrons are free to replace lost electrons. As the photon absorption process continues, those electrons lost by P680 are transported by special carrier molecules to the electron-deficient P700. The electrons lost by P700 are also passed on by carrier molecules to end up ultimately outside the thylakoid in a region called the stroma. This process of moving electrons from water to P680 to P700 and into the stroma is called the electron transport system. The electron transport system consists of an elaborate chain-like series of oxidation-reduction reactions. For our purposes, we'll think of the oxidation-reduction reactions in their simplest form. Oxidation means losing electrons, while reduction, the opposite process, means gaining electrons. Both oxidation and reduction must occur in order for the electron transport system to function. Each time one of the electron carrier molecules loses an electron, it is said to be oxidized. And the electron carrier that gains the electron is reduced. While the main purpose of the electron transport system is to move electrons through the photosynthetic unit, some of the carrier molecules perform an additional function. That is, they transport both electrons and protons in the form of hydrogen atoms. Because of this extra function, they are called hydrogen carriers. One of these special hydrogen carriers, quinone, or PQ, located between photosystem 680 and photosystem 700, acts as a proton pump between the stroma and the thylakoid interior. The protons lie outside the thylakoid in the stroma, and the role of PQ, as well as transporting electrons, is to transfer protons from the stroma down to the thylakoid interior. However, PQ can't carry naked protons. It can only carry hydrogen atoms. So it overcomes this dilemma by picking up two electrons from the transport system protons from the stroma and combines them to form two hydrogen atoms. PQ, having acquired the two hydrogen atoms, has gained electrons and has therefore been reduced. The reduced PQ is called PQH2. The H2 signifies that it is indeed carrying two hydrogen atoms. Now the unique ability of plastoquinone comes into play. It physically moves or diffuses down through the thylakoid membrane to its inner boundary. Where it releases the two protons into the thylakoid interior and the two electrons back into the transport system and on their way to electron-deficient photosystem 700. Freed of its burden, the plastoquinone returns to start the process all over again. Beyond photosystem 700 are other electron carriers in the transport system. Energy-rich electrons from P700 move from one carrier to the next, until they arrive at the small carrier molecule, FAD, at the end of the electron transport system. Once FAD acquires two electrons, a molecule of NADP moves into position to pick them up. But NADP is a hydrogen carrier. So at the instant the electrons are passed from FAD to NADP, two protons from the stroma combine with the electrons to form two complete hydrogen atoms. Having gained electrons by acquiring two hydrogen atoms, the NADP has been reduced and is now called NADPH2. The reduced NADPH2 moves back up into the stroma. So, hydrogen carriers perform two separate tasks: the transport of electrons and protons. The protons released by the plastoquinone mingle with the protons liberated during the photolysis of water, gradually increasing the concentration of protons in the thylakoid interior. These protons play a critically important role in photosynthesis. As the proton buildup continues, pressure is relieved with the removal of surplus protons through special ion channels in the membrane. At the outer end of each channel is a large complex of enzymes called a CF1 particle. As the protons pass into the CF1 particle, it taps some of their energy to convert adenosine diphosphate, ADP, to adenosine triphosphate, or ATP. The whole of the light reaction has led to this, the production of ATP. ATP is so important that biologists refer to it as the power supply of life.